Hello class, and welcome to our fourth lecture video here on Unit 6, Cell Division and DNA Replication. In this lecture video, we're going to begin to discuss the basic structure of DNA, which again stands for deoxyribonucleic acid. So let's get started. So to begin, let's give a basic definition and function again of what DNA is. So it is that molecule that contains all of the genetic code of an organism. Every organism on Earth has this information. And what the information really is simply directions on how to assemble proteins. So inside this DNA molecule over here moving around, and again, this is called the double helix, right? Because you have two sides of the DNA that are connected in the middle, and they kind of create a helical shape. The information within that DNA really is just which amino acids do I need to put in order to create a particular protein. Now this big huge molecule here, its monomer units, if you recall from our second unit this year, are nucleotides, and here they are down here. Here's the basic structure of one nucleotide. And each and every nucleotide is made up of 3 parts. Again, we did mention this earlier this year in biochemistry. We first have a 5-carbon sugar, and in regards to DNA, that's going to be deoxyribose. Right? It's a 5-carbon sugar. It's always going to be the one that you see in the middle of the overall molecule, or monomer unit, I could say. And in regards to the carbons, I will come back to this in a moment, but I would number them as such. So right here where my laser pointer is, this would be the 1st carbon, 2nd carbon, 3rd carbon, 4th carbon, and then here's the 5th carbon of deoxyribose. All right, this is just an oxygen that bridges between these 2 carbons. A phosphate group always is attached to the 5th carbon of deoxyribose, and one of 4 possible nitrogen-containing bases, which is right here, is always attached to the 1st carbon in deoxyribose. It's easy to discern which one is which. There's a phosphate in the phosphate group, and the nitrogen-containing base always has a bunch of nitrogens within the molecule. You can see itself also. And again, deoxyribose would be here in the middle. So you have billions of these monomer units that are all linked together to create this overall double helical structure that you see moving around. Now let's expand on the possible nitrogen bases here. There are in all DNA, so for any organism on Earth, I don't care if it's chimpanzee, baboon, human, COVID, all organisms, all structures that use DNA will use 4 possible nitrogen-containing bases. And they are as follows, in no particular order. We have adenine and guanine, and then cytosine and thymine. The way these nitrogen bases are broken down is into 2 classes. Adenine and guanine, or you can just simply say A and G. I'm never going to have you write out the actual names of them. I just want you to recognize them if you see them written like that. So adenine and guanine, or A and G, are considered to be purines. Now what's that significance? They are a double ring structure that you can see up here above. Alright, so you have 2 carbon rings with nitrogens mixed in, connected together. So the way I always remember it is smaller word purine, larger overall structure. So A and G are what we call purines. C and T, or T and C, thymine and cytosine, are considered to be pyrimidines. Larger word, smaller structure. They each are just a single ring compared to the double rings. Alright, so I want you to remember which class, purines or pyrimidines, each one of these 4 bases falls into. Now, if you want to understand all life on Earth, you just need to know some simple base pairing rules. In all DNA, we always see the following base pairs occurring. We're always going to see adenine pairing up with thymine. So that means we have a purine binding with a pyrimidine. And then cytosine always pairs with guanine, or you could say guanine pairs with cytosine, either way, which again is a purine binding with the pyrimidine. So A with T and G with C. All life on Earth, all life that we know of that exists, follows these two simplistic base pairing rules. To visualize it, we can look at it like this. So in this section here, what we have is adenine on the right. It's a double ring structure binding with thymine. And over here we have guanine, again double ring structure binding with cytosine. Now the reason why we have these base pairing rules is based on the number of hydrogen bonds each of these bases can form. Adenine and thymine together can only form 2 hydrogen bonds. So there's those dotted lines that should look familiar when we did basic chemistry and bonding earlier this year. Again, it's just a weak attractive force between either an oxygen or nitrogen, all right, with a hydrogen on another molecule. So you can see that going back and forth. While guanine and cytosine, they can form up to 3 hydrogen bonds. So this is more stable than just 2. So that's why G and C always prefer to bind with each other. Is it possible that guanine could bind with thymine? Yes, but that would be considered a mutation and an error in our DNA, and we don't want that. So this is what we always want to see. So inside a DNA molecule, inside the DNA ladder, the 2 sides of DNA are simply being held together by the weakest bond on Earth, which again are hydrogen bonds. Now, the different order of bases is what really differentiates one organism from another. So what do I mean by that? So again, here let's pretend is one-sided DNA. All right, so we're just looking at one half of DNA. So I have A, T, G, C, T, T, etc. down the line, right? Well, if I only had one-sided DNA, that's really all I need in order to create what the other double-stranded side of DNA should be, or what we call the complementary side of DNA. All I got to do is follow the base pairing rules. So if I have an A on one side, I should have T on the other. If I have T on one side, I should have A on the other. G on one side, C on the other. And that would continue until you create complementary base pairing between both sides of the DNA strand. So you could do this for any organism on Earth. Again, another example, if you had one-sided DNA, and if I just look at the first 2 sets of bases here, TTT, CTA, follow your rules, A with T, G with C, and the base pairs should always match up all along the strand. So it doesn't matter which side of DNA you have, if you know one side, you could theoretically recreate the other side to create your complementary double helix. So based on the sequence or order of these nitrogen bases, that allows you to figure out how closely related you are to other organisms out there in the world. So for example, humans, their closest living relative based on these sequence of bases that we've been able to discover are in fact chimpanzees, which fall underneath the primate category, right? The great apes like you and I are. After that, DNA changes a little bit, but gorillas would be our next closest living relative or cousin. And then after that, you'd have orangutans. And you would keep going down after that to all other great apes, but then you would move into monkeys, then you move into other species as well. All right? So again, the similarity of these bases, the sequence of them, is what ultimately relates you to other organisms on Earth and how long it's been since you shared a common ancestor evolutionary-wise with them. All right, so we'll come back to looking at DNA sequences when we, towards the end of the year, we get into evolution. Looking again at the structure here, I just want to point something out to you. Here's that deoxyribose. Again, we numbered the carbons earlier, right? 1, 2, 3, 4, 5. The 5th carbon is where a phosphate group binds. The 1st is where one of those 4 nitrogen bases bind. So all DNA molecules, every double helix, is really made up of 2 strands of nucleotides. So highlighted in blue are the 2 sides of DNA. If I were to circle one nucleotide for you right now with my laser pointer, that's what I'm doing right now. So I'm circling one base, one sugar, one phosphate group. So each side of DNA is just made up of a long strand of nucleotides, And then the two strands of DNA are connected in the middle between their base pairs by those weak hydrogen bonds. So all other bonds in DNA, especially between the sugars and phosphate groups, which make up the backbone of DNA, like the spine, are covalent bonds, those solid lines between phosphates and the carbons of the other sugars. The hydrogen bonds are what ultimately just connect the two sides. Those hydrogen bonds are very easy to break, which is a good thing because it allows me to unwind and open up the DNA when I need to gain access to the code. Bad because these bonds can break fairly easily, which can lead to mutations and aging and disease in life. But the benefits outweigh the negatives right now to allow you to live your life for a decent amount of time. Now, lastly, what I want to bring to your attention with the structure, because this is going to help us understand DNA replication a little bit better later on, is that the two sides of DNA run anti-parallel to each other. So again, parallel means right next to each other, right? But in order to have the bases bind together, you have to change the direction in which the nucleotides run. So if I look over here, because I think it's a little bit easier to see here in this diagram, here's the deoxyribose of one nucleotide, and here's the deoxyribose of another. Notice that the oxygen is facing up, like you see down here on this side, but the oxygen is facing down on the other side. That's what I mean by anti-parallel. You have to flip the nucleotides upside down and spin them around in order to get these base pairs to match up. So on what we call the 5' end of DNA, that's when the 5th carbon of deoxyribose is facing upwards. So if you look down, that means the 3rd carbon on deoxyribose, so right here where my laser pointer is compared to here, is facing in the downward direction. Well, on the other side of DNA, in the upwards direction, right here where my laser pointer is the 3' end, or the 3rd carbon in deoxyribose is facing upwards. And then facing downwards, right here, would be the 5th carbon, which we said then is attached to a phosphate group. So realize that the two sides of DNA, in order to get the base pairs matching up, they have to run in opposite directions or anti-parallel to each other. That's going to affect the rate and speed in which each side of DNA can be duplicated later on in DNA replication. So we'll rehash this and come back to it in a little while. To finish out this lecture video, I just want to go over what the definition of a gene is. All right, because genes are on chromosomes that we've already addressed. Again, chromosomes are made of DNA. All right, just condensed chromatin. But on every chromosome, you're going to have sections of DNA called the gene. All right, and here's those banding patterns that we could see on chromosomes we discussed earlier, right? Those are genes. So in every chromosome, you could have hundreds, thousands of genes. And what a gene is ultimately is this a section of DNA or a section of a chromosome that has the instructions on how to make a very specific protein. So again, as I mentioned earlier, those sequence of nitrogen bases, the A's, the T's, the C's, and the G's, are really just the instructions on how to put amino acids together to make a particular protein. All right, and as proteins change based on the change in bases, that can ultimately alter or change how the body of an organism is adjusted. All right, between a human being and, let's say, a fruit fly. We both have similar genes, but as those bases get altered over time due to mutations, it could ultimately lead to differences in how body parts, proteins, your immune system are created and how they function. All right. When we get more into genes and genetics, that'll be directly related to protein synthesis.